welcome to Bio40 Pre-Lecture 7. We're going to spend the next two lectures discussing how we can map the relative positions of genes along chromosomes. We talked earlier about how the movement of chromosomes during meiosis explains how independent assortment works with genes on different chromosomes. A purple chromosome with the dominant A allele has an equal chance of segregating with a blue chromosome that has a dominant B allele or a recessive one. So all four combinations of gametes shown here are possible. But what about genes on the same chromosome? After all, there are many genes on each chromosome. Currently, we estimate humans have around 22,000 genes, which means an average of nearly 1,000 genes per chromosome. Our largest chromosomes actually have several thousand genes, while our smallest have just several dozen. So do all the genes on the same chromosome segregate together in every meiosis? If that were the case, genes on the same chromosome would always be passed through generations as a block. Such traits would be inseparably linked. Well, you probably remember that we also talked about why that's not necessarily the case. In prophase 1, the homologous chromosomes pair, and pieces of non-sister chromatids physically break apart, exchange positions, and reattach. And this happens at least once for each tetrad per meiosis, though the location of the crossover is mostly random. Scientists had long thought such events were likely to occur, but physical confirmation did not arrive until the work of Barbara McClintock and Harriet Crichton in the 1930s, a good 40 years after chromosomes were first observed. So crossing over makes independent assortment of genes in the same chromosome possible. If genes A and B are on opposite ends of a chromosome, you can have a crossing over event between them, and this will lead to the formation of gametes with all four potential arrangements of alleles. Remember, we call the chromosomes with the same configurations of alleles as the parents, parental, and the others, recombinant. Now what if the genes A and B weren't so far apart? Wouldn't it become less and less likely that a crossover occurs between them? Well, yes. So the principle of independent assortment breaks down specifically for genes that are close together on the same chromosome. The probability of each potential gamete forming is no longer equal here. Rather, the parental gametes become more probable than the recombinant. Luckily in science, when a supposed rule fails, this isn't a problem, but rather an opportunity to learn more about the underlying complexities. In this case, we can use the knowledge that distance affects assortment to determine the order and proximity of genes along a chromosome. If we're going to relate recombination to mapping, the first thing we need to do is develop a method for calculating the percentage of recombinant gametes. To do that, we need several things. We need to start with a dihybrid parent, someone who has one wild type and one mutant allele for both of the two genes you're looking at. We also need to know the linkage relationship. If the wild type allele of each gene is on one chromosome and the mutant's on the other, the genes are said to be in coupling. When each chromosome has one wild type allele and one mutant, the genes are in repulsion. After a crossover event, the linkage relationship in the recombinant gametes will be different from that of the parents. But this is difficult to observe directly. What we want to do is figure out an informative cross that allows us to infer this information by looking at the offspring. First, we'll look at how to do such a cross when dealing with X-linked traits. Here's an example with fruit flies. We already talked about the gene for eye color being X-linked, and another X-linked trait is body color. Flies with a wild type Y+ allele have typical tan bodies, but those homozygous for the recessive Y allele are yellow. So let's cross a true breeding white-eyed tan female with a red-eyed yellow male. All the F1 females will have a wild-type phenotype, but we know that they are heterozygous for both genes. We have our dihybrids. Now for the linkage relationship. We know the mother must have passed on a mutant W allele and a wild-type Y+ allele on one X chromosome, while the father contributed a wild-type W+ allele and mutant Y on the other. The genes are in repulsion. So who do we cross the dihybrid females with, and how do we identify parental and recombinant chromosomes in the offspring? Well, it ends up we can cross the female with any male, which means the easiest thing to do is just let the F1 siblings mate. The reason the mating partner doesn't matter is that we're just going to look at the males in the F2. Because the F2 males will always inherit a Y chromosome from the father, we don't care about the father's X chromosome. Always inheriting a Y chromosome from the father also means that the mother's alleles will always be readily apparent in the F2 males. White-eyed tan males must have inherited a W Y+ chromosome from the mother. Red-eyed yellow males must have gotten a W+ Y chromosome, and so on. The phenotypes of the F2 females don't tell us anything, though. The Y plus allele from the father's X chromosome means all the daughters will be tan, so we can't tell what allele was inherited from the mother. But with the males, we can tell who inherited a parental chromosome and who inherited a recombinant. We already established the F1 females had WY plus and W plus Y chromosomes, so we know the sons with one of those chromosomes are parental types, while the others are recombinant types. It's important to note that the designation of parental and recombinant are based on the specific cross we're talking about and are subject to change from one situation to the next. If instead of the situation we described, the F1 female had W plus Y plus and WY chromosomes, those would be the parental classes, and W plus Y and WY plus would be recombinant. Don't make the mistake of thinking coupling is always parental or anything like that. Getting back to the cross we started with, now that we know which offspring are parental types and which are recombinant, we can calculate the percentage of each by dividing the total of each class by the overall total number of offspring. We tend to focus on the percentage of recombinants, which is often called the recombination frequency. Here we see the recombination frequency is about 1%, far from what you'd expect with independent assortment. Indeed, when you look at genes on different chromosomes, the recombination frequency will be 50%, allowing a little error due to sample size. The chromosomes segregate independently, so all 4 potential gametes are equally likely, and the recombinant progeny will be as common as parental. One important thing to note is the nomenclature we use when dealing with genes on multiple chromosomes. A semicolon is used to separate genes known to be on different chromosomes. Contrast that to the nomenclature we use for genes on one chromosome. Here, we don't use a semicolon and put each gene on the same side of the slash. So a slash separates alleles of a single chromosome, while semicolons separate different chromosomes. Here's an example of 2 genes that are far apart on the single chromosome. As we've said, when genes are far enough apart, there will be a crossover event between them nearly every meiosis. This makes the number of parental and recombinant chromosomes nearly even, which is reflected in the offspring. There is no situation where the number of recombinant offspring should be greater than the number of parental. If there is no crossover event between 2 genes, all 4 gametes will be parental. With one crossover event, there will be 2 parental and 2 recombinant gametes. But I've suggested before that there can be multiple crossover events per meiosis. What if a second event occurs between the other set of sister chromatids? In this case, all 4 gametes will be recombinant. The thing to consider here is that it's equally likely that the second crossover will be between the same chromatids as the first. And in this case, we've reset back to 4 parentals. So on average, the number of parental and recombinant gametes will still be equal. It's also worth noting that the second crossover can be between one chromatid that was involved in the first crossover and one new one. But this just gives an equal number of parental and recombinant chromosomes. This slide is a good summary of the situations we just looked at. Again, a double crossover can create a situation where there are only recombinant gametes, but on average, the parental and recombinants will be equal. Now it's time to talk about how we use recombination frequencies. A.H. Sturtevant reasoned that the percent of recombinants should reflect a physical distance between 2 genes. He set a recombination frequency of 1% as 1 map unit, which you also see referred to as a centimorgan. So if there are 1.1% recombinant offspring from an individual heterozygous for W and Y, we say that W and Y are 1.1 map units apart. If there are 32.8 map units between W and M, that means that 32.8% of the offspring of a WM dihybrid were recombinant. These units are somewhat arbitrary, but they help us understand relative positions. We can tell that W is much closer to Y than it is to M. With enough crosses, we can map the relative position and distances between many genes. This will be an exercise we'll save for class. A group of genes that can be linked together by such crosses are called linkage groups. And if you look at enough genes, linkage groups actually reveal all the genes that are on a chromosome. If you look at the gene that causes fruit flies to have yellow bodies, you'll see it's too far from the gene that causes carnation eye color to be linked directly. Those genes will appear to assort independently. But the yellow body gene is linked to the singed bristle gene, which in turn is linked to the rudimentary wing gene, and that is linked to the carnation eye gene. So that is how we can put all of these genes along the chromosome into a single linkage group, even though some pairs of genes are more than 50 map units apart. These sorts of maps are good at telling us the order of genes, but there are some caveats to consider when relating these recombination frequencies to physical distances. For one, a map unit can mean very different things in different organisms. Later, when we establish DNA as the important molecule in chromosomes, we'll talk about distance in terms of base pairs, or BP. And a map unit in humans averages about 1 million base pairs, compared to a map unit in yeast of merely 2,500 base pairs. Even looking at just a single organism, there can be variations in recombination rate along a chromosome. If there's a hotspot, a place where recombination is particularly common between two genes, you will see a higher recombination frequency and the genes will appear closer together than they are. Finally, recombination rates can vary between the sexes. In the most extreme example, male Drosophila don't do any recombination. That's all for this week. Next week we'll do some more complicated crosses that will get us some more accurate maps. Also, have I not explained how to get recombination frequencies for autosomal genes?